Enzyme assay for safety indicators. Enzyme-linked immunoabsorbent assay is one of the most common assays used for detection of pathogens such as Salmonella, Listeria monocytogenes, E. coli, and Campylobacter jejuni, or their toxins. It involves three basic steps. First is the immune reaction, which is reaction between the antibody and antigen. Second is the enzymatic chemical reaction, which is the reaction between the enzyme and the substrate, which will produce a colored compound. Third is the signal detection and quantification, that is, detection and measurement of color intensity of the colored product generated by the enzyme and added substrate. Now, the basic principle involved in this assay. Immunoglobulins, also known as antibodies, are manufactured protein by the immune system to help fighting against the foreign substances. Any foreign element that causes the immune system to respond to their presence by producing antibodies can be considered as an antigen. Antigens are living organisms from a variety of families including viruses, bacteria, fungi, chemicals, pollen grains, or even food allergens. There exist 5 different antibodies types with their specific configuration and functions. These antibodies are IgG, IgA, IgM, IgD, and IgE. Now that we know what is antibody and antigen, we will go deep in their principles. Antigen and antibody coupling. Antibodies mainly possesses Y shapes with different upper branches of the Y. Due to their specific amino acids, antibodies are able to identify specific types of antigens from the binding sites that exist on the surface of antigens. Through an immunological response to the presence of an antigen, the antibody wraps its two arms, the two arms represent the Y shape of the antibody, around the antigen's combining sites and capture the foreign agent via the lock and key correlation to stop its progress. Specificity of the antigen-antibody coupling. In the structure of antibodies, there are genes that direct the construction of specific sites for binding to the antigens. Such antigen-specific regions are located at the end of the Y-shaped arm of antibodies. In the case of toxin antigens produced by pathogenic bacteria, the antibody-antigen coupling process occurs along with neutralization of the antigen's toxin component. Biomolecular interaction between antibody and antigen. The major forces involved in between antibody and antigens are hydrogen bonding, ionic attraction, hydrophobic interactions, and van der Waals forces. Enzyme-linked immunosorbent assay. Conducting an ELISA includes at least one antibody with specific immune response against the antigen of interest, which means the antigen we are detecting. The detectable agents, which can be antigens or antibodies, can be immobilized on supporting substrate either through direct non-specific adsorption or by specific capturing by another antibodies, which we see in sandwich ELISA. After immobilization of antigen, the surface is normally coated with a blocking agent to reduce the chances of non-specific bonding in the next steps of the assay. The labeled antibodies is added and subsequently coupled with the antigen, forming a complex that leads to the detection of signal. The labeled antibody can be enzyme linked through a covalent method or can be further detected by a bioconjugated secondary antibody. Between each step, the plate is thoroughly washed with a washing buffer, that is a mild detergent solution, to remove unbound proteins without leaving any negative effect on the bound proteins. The substrate is then added to the plate to develop the enzymatic reaction and to produce the color from which The signal intensity can be measured through spectrophotometer. Elements of the assay. Solid phase. This is well-known platform includes microtiter well plates, small balls and small tubes. The 96 well plates are typically made from polystyrene, which is cost-effective, highly transparent, and relatively hydrophilic material suitable for protein adsorption. Solid support can also be available in the pre-coated form containing specific types of antibodies or antigen present on the surface. Adsorbents. Antibodies and antigens are the main adsorbent used in ELISA. Target biomolecules, which are the biomolecules which can be detected by ELISA, would include human antibodies, DNAs, RNAs, hormones, tumor markers, genetically modified organisms, infectious agents, as well as air and food allergens. Washing agents. By charging and emptying the wells with specific amount of washing buffer, unreacted reagents can be removed from the wells. Hence, the accuracy of the assay increases as only reacted analytes remains in the well plates. Typical washing buffer consists of small concentration of non-ionic detergents such as twin 20 in phosphate buffer saline solutions. Enzyme and substrates. The enzyme chemically modifies the substrate into a measurable product, thus signal readout is possible. For every class of enzyme, a specific type of substrate is required. Commonly used enzymes are horseradish peroxidase and alkaline phosphatase. Upon addition of substrates such as O-phenyldiamine and tetramethylbenzidine, respectively, a positive color reaction is quantified by measuring the color intensity with a spectrophotometer. Types of ELISA. There are majorly four types of ELISA: direct ELISA, indirect ELISA, sandwich, and competitive. Starting with direct ELISA. The first step involves attachment of the antibody of interest to the solid surface of the well. Then it will be washed. After rinsing to remove excess antibodies, sample with suspected antigen is added. In the following step, an enzyme labeled primary antibody is introduced to the assay. Colorless substrate is added and development of color determine the presence of antigens. Indirect ELISA. In indirect ELISA, the antigen is first immobilized on the polystyrene plate and reacted with a primary antibody, which is opposite to direct ELISA, in which the antibody is first immobilized. Then, after washing, an enzyme labeled secondary antibody is added and then allowed to bind with primary antibody. Then a substrate solution is added for development of color. Sandwich ELISA. Sandwich assay, as its name suggests, wraps the analyte of interest in between the primary and secondary antibodies from both sides. In sandwich ELISA, an antibody is first immobilized on the plate and is then allowed to capture the antigens. The primary antibody, which is immobilized, is also sometimes called capture antibody. Then a second pathogenic antibody labeled with the enzyme is added, forming a typical sandwich configuration. Antigen is required to have at least two binding sites for antibodies. As we can see here, the antigen is the yellow, which is captured or sandwiched in between the capture antibody and the secondary antibody, having an enzyme which will react with substrate and produce a colored compound, which then can be quantified in here. Here is the diagrammatic representation of all the different types of ELISAs. First is the indirect ELISA, in which we can see the antigen is immobilized with the plate or the well. This is the primary antibodies which is need to be detected in the sample. This is the secondary antibodies which is linked with enzyme which will react with the substrate and produce color. Now this is sandwich ELISA. You can see this antigen is actually sandwiched in between the primary or capture antibody and the secondary or enzyme linked antibody. Third, competitive ELISA. In competitive inhibition ELISA, bacterial cells or antigens are first immobilized in the well. In separate tubes, the primary antibodies is mixed with various dilution of bacteria and added to the wells. Now, the previously mixed antibodies and antigens are added to the wells. Only those antibodies which are not binded with the antigens will attacked to the antigen immobilized on the well. Enzyme linked with antibodies will be added further after the addition of the primary antibody and bacterial solution, and then the substrate will be added upon which the color reaction will happen with the enzyme and color will be produced. These are the references I have used. Thank you.